Yeah, I think it started recording. Yes, it started recording. So, welcome to the day 11 session on concepts of reverse vaccinology in vaccine design. So, before proceeding to today's topics, can we recap the last session topics? So, we discussed something on QSCR, isn't it? We have discussed about the dimensions of QSCR, and we discussed something on training data set model, test data set model, etc. And then we have discussed what is QSPR, QSTR, and then we have seen how to go with the pharmacophore mapping using form mapper. Okay, so this is what we have discussed, and I also showed you the results how it looks like in form mapper. Now let me take you to the today's topics, that is on immunoinformatics reverse vaccinology. So what is immunoinformatics, and how about the different tools and databases used in immunoinformatics we'll be discussing here, and about vaccine adjuvants, types of vaccines, and what is reverse vaccinology, and case studies in reverse vaccinology. I'll be circulating a journal paper published on reverse vaccinology, so you can also go through the results and the indigenous database, whatever developed in Sri Venkateswara College of Engineering. And then we discuss on epitope databases. Two types of epitope databases available, B cell and T cell. We'll be discussing about this information on today. First, about immunoinformatics. Immunoinformatics, also called as computational immunology. Am I audible to everyone? Yes or no? Yes? No? Any response I can get from your side?
Yes, sir. Yes, sir.
Yes, madam. Thank you, ma'am. So let me continue. So I was discussing something on immunoinformatics, which is also called as computational immunology. This is a field of science that encompasses high throughput genomic and bioinformatics approaches to immunology. Okay, and whatever we discussed earlier, computational biology. Okay, the related to drug design, molecular mechanics. That is, molecular mechanics is basically a biophysics subject, isn't it? So whatever we discussed earlier, that is actually related to molecular biology. That is related to proteins, related to DNA, RNA. But whereas here, this is something different. This is something related to computational immunology. Okay, biology there we mean about molecular biology in default. Now let me take you something about the definitions of vaccine and vaccine adjuvant. What is vaccine? It is a biological preparation that induces acquired immunity against a particular pathogen. For example, if a person gets wounded, then obviously we can able to get two types of responses, immune responses will be acquiring. One is actually a kind of a humoral response. Whatever I am telling, a humoral response, that is humoral immunity, occurred through antibodies. You know, antibodies are available, isn't it, in our body, which are produced by B cells. Okay, B cells only produce the antibodies. If you see, the humoral response is acquired through the antibodies produced from B cells. And there is one more immunity called cellular immunity. In cellular immunity, the B cells are generally considered as antigen presenting cells. It means that on the surface of a surface receptor called major histocompatibility complex. Okay, the name of surface receptor is major histocompatibility complex. In that surface protein, we can able to see and antigen is present. Okay, the antigen can be a viral antigen or bacterial antigen, whatever bacteria or virus getting inside the cells can um, degrade the membrane and it can release the particles. And these particles, the antigen particles, are processed and presented on the surface of MHC molecules. Okay, and if we have this antigen from any of the foreign source, that can be a bacteria or virus, if it interacts with T cells, Okay, there are another group of cells called as T cells. Okay, the T cells generally, the T cells generally have a kind of another receptors. The receptors present in T cell try to interact with the antigen of B cells. Okay, this is what we call it as cellular immunity. In cellular immunity, you can able to see that T cells interacts with the B cells and other antigen presenting cells. Like B cells, we have antigen presenting cells like dendritic cells, okay, and macrophages. So these are examples actually, macrophages, dendritic cells. So these have MHC molecule on the surface, and the function of antigen presenting cells is to present, not only just present on the surfaces, but to interact with the T cell receptors. Okay, if it interacts with the T cell receptors, we can see that the T cell gets activated, and one T cell is going to proliferate into two T cells. So this is how we can able to find. And not only that, these T cells can also produce cytokines. The cytokines are proteins. These proteins in turn can interact with the antigen presenting cells, and this can also activate APCs, that is antigen presenting cells. So these types of immunity, humoral and cellular immunity, together we call it as overall immunity. Okay, and if you see this vaccine, in vaccine, whatever biological preparation, the biological preparation can be, if you want to treat a viral infection, we can use the dead virus, or we can use the treated virus, or even we can use the viral particles. So any of this can be used as a vaccine to combat that kind of particular pathogen. Okay, so this is actually used for prevention of disease instead of treatment of disease. For treatment of disease, we generally take drug. But if you want to uh, overcome the infections in future, okay, with the bacteria or virus, we can able to take the vaccine, which is nothing but a biological preparation, where you can obtain these types of immunity. Okay, and if you see in another term called adjuvant, which is a component of vaccine. The purpose of adjuvant is if you see an adjuvant, generally it is a chemical or a biological agent, okay, and that is added to a vaccine. As I mentioned, this is a component of vaccine. So the purpose of adding this adjuvant to the vaccine is to boost the humoral or cellular immune response. Whatever I told, the B cells alone produce proteins, the soluble proteins called antibodies. Similarly, the B cell can interact with T cell to form few more T cells, and even B cell gets activated. This is what we call it as cellular immune response. So the objective of using the adjuvant is just to increase the efficacy of antigen. If you want to prolong the vaccine period, for example, if you take BCG, you know what is the lifespan of BCG? Like before going to BCG, I can ask you simple questions. If we take vaccines like Covishield or Covaxin, this is what we have taken, isn't it? Recently, BCG we might have taken in the childhood. How about the Covishield and Covaxin? I think you have an idea on this. What is the lifespan of this vaccine? If you take a vaccine on today, how many years or how many days or how many months we can able to protect from that particular SARS-CoV-2? Can you guess what is the shell life or lifespan? How much lifespan we can able to anticipate from this?
2 years, sir.
Very good, ma'am. Nice try from your side. So approximately 1 year. So if it is 2 years, then it will be a best, I believe. Well, you are right. The lifespan is very limited actually, not more than 2 years. Okay, and if you see the other vaccines like BCG, okay, this can be used up to even 10 years or more than 10 years also. Okay, I just mentioned the minimum. So maximum we can take to the lifespan. Okay. So like this, we can able to anticipate the shell life varies with respect to vaccine to vaccine. If so, to increase that shell life, okay, we can use this chemical or biological component. That is what we call it as adjuvant. Efficacy, a term newly I've introduced. Like how we can able to measure the performance of a ceiling fan through efficiency. Similarly, the performance of vaccine antigen can be, or the potency can be measured through efficacy. This is the generic term and terminology we use to measure the response, immune response. If you see the vaccine adjuvants, okay, the properties of vaccine adjuvants is to produce high antibodies. As I mentioned, the only cell which produces antibodies are B cells, B lymphocytes. The next thing is to make antigen more immunogenic. If only it creates more immunity, then it is more applicable for treatment or for prevention of disease, isn't it? And similarly, generation of specific cytotoxic T cells and memory T cells. So these are the major applications of vaccine adjuvants. Now let me take you to the types of vaccines. If you see the types of vaccines, we have inactivated vaccines like hepatitis A, flu, polio, rabies, live attenuated vaccines, measles, mumps. What is the difference between inactivated, live attenuated? You have any idea? Inactivated, also called as killed, isn't it? And live attenuated. Actually, we use heat treatment or chemical treatment, okay, to make the virulence, to make null virulence, to remove the virulence of the virus. We either subject to heat treatment or chemical treatment. Okay, so because of that, whatever uh, virus we want to treat, viral infection we want to prevent, we can able to prevent from this. Okay, so either using the killed viruses like hepatitis virus, flu virus, polio virus, we can able to prevent this kind of viral infection, or else even we can use the treated kind of vaccines. Okay, heat or chemical treatment. And now let me take you to the third thing is the messenger RNA vaccines. Messenger RNA vaccines we have heard, isn't it, for COVID-19, for SARS-CoV-2 we have so many types of vaccines. One such type of vaccine is messenger RNA vaccines. The fourth is the subunit. The subunit is nothing but a part of a virus. Like it can be a membrane or even it can be a protein present inside the virus. Okay, so the protein part present in the viruses, not only the protein. Here they have given another example like polysaccharide. So subunit, recombinant type of vaccines, or even polysaccharide vaccines, and conjugate vaccines. Okay, everything comes under another category, like hepatitis B, human papillomavirus, meningococcal disease. This is generally happens in brain. Okay, for all these things we can use this subunit type of vaccines. The fifth one is the toxoid. For example, diphtheria and tetanus viruses can produce toxins, and these toxins can able to prevent the infection from these viruses. That's what we call it as toxoid vaccines. The sixth is the viral vector vaccines. So from adenovirus vectors, and we have so many types of viral vectors. Okay, popularly from adenovirus, we can able to take the vaccines, isn't it, for COVID-19? And the seventh is the peptide-based vaccines. In peptide-based vaccines, the example provided for treatment of COVID-19 is COVAX, which is nothing but a multitome-based vaccine. Now let me take you to the major concept, that is how to distinguish classical vaccinology and reverse vaccinology. So till now we have discussed what is immunoinformatics, the immunoinformatics, nothing but computational biology. Then we have discussed something on vaccines, definition of vaccine and vaccine adjuvant. Then we came to know the biological properties of the vaccine adjuvant, and then we came to the types of vaccines. Now we are discussing on classical vaccinology. If you see the classical vaccinology, okay, as I mentioned earlier in the previous slide, the vaccines are manufactured directly from the biological component, that is, directly from the virus or directly from the bacteria or even from the parasite. So, whatever we just grow the viruses or bacterium either killed or we treated with a chemical or heat, okay, in a bioreactors, whatever the reactors used to grow these kind of cultures, bacterial or viral cultures, later this is actually manufactured in the form of vaccines in the form of vials. But if you take the reverse vaccinology, okay, so we know that the genes, proteins of viruses are already discovered. So from this, even from the genes, from the chromosomes, so all the genome is also constructed. At present, for most of the species, human, animal species, bacteria, virus, plants, all these genomes are sequenced. So if you want to work on the production of SARS-CoV-2 vaccine, that is for the treatment of COVID-19 disease, then obviously we can start with even genome. What is the purpose why we have to go with the once again with SARS-CoV-2? In SARS-CoV-2, we have 100 types of mutations. Whatever we have taken Covishield and Covaxin, it can able to prevent only the first strain of SARS-CoV-2 virus. This can't be able to prevent other strains. We have strains like alpha, beta, gamma, delta, so many things. Okay, so many strains are available. If you see this delta strain, if you tell that alpha strain is originated from Wuhan, that's what the literature papers provide the information. The delta virus originated from India. How many know this? Similarly, even one or two strains are also originated in India from Indian bands. Okay, if you see that, actually in the reverse vaccinology, we start with the genome. Okay, that each and every strain, whatever I mentioned, 100 strains. Okay, just because of a single mutation in spike glycoprotein, it leads to another different strain. So, whatever vaccines we use initially for treatment of alpha can't be able to use for the prevention of delta strain. The if you see the efficacy of Covishield and Covaxin with respect to Delta, it comes less than 20. Okay, so that is only 20% efficacy. 80% people won't get prevented from this Delta strains. So then what we have to do, we have to come with another new vaccine for the specific Delta strain. Okay, so this is what we call it as precision medicine approach. So in our course, in our internship and certificate course, also provide you hands-on to specifically target the people. For example, um, for the more examples we see in the subsequent slides. Now let me complete the reverse vaccinology. In the reverse vaccinology, already the genomes are established. Okay, so from genome, proteome, interactome, metabolome, we take this kind of bioinformatics data, including the structure data of proteins. Okay, and if we take this data, am I audible? I'm getting so many messages. I think it's not related to ours. Am I audible to everyone? Yes? No?
Sir.
Uh, okay. So is that my internet frequency is good? I mean, you are getting continuous voice from my side?
Yes, sir. Yes, sir.
Okay, ma'am. Thank you. Thank you. Thank you, ma'am. So now I was discussing something on reverse vaccinology. Okay, but no, I just scared like whether I came out of from the internet or not. So when I'm just confirming, getting confirmation in between. Now let me go ahead. So genome, proteome, interactome, metabolome, structure of proteins, everything are useful to come up directly with a kind of peptide-based vaccine or RNA-based vaccine or even any uh, even a subunit-based vaccine. Okay, so all these things we come up with the already generated data. So that's what we call it as reverse vaccinology. Instead of coming from the source, coming from the virus or bacteria or parasite, we are taking from these bioinformatics data. Okay. And now something about the pipeline of reverse vaccinology, how it works. We just start with the genome sequence and we go with the computational analysis. And we initially try to predict two types of epitopes, that is B cell epitope and T cell epitope. And whatever epitopes I mentioned are nothing but peptides. Peptides, whatever we try to use, is around 9 to 13 amino acid peptides. Some can also go up to 15 amino acid peptides. This is the length of peptide generally used to prevent the infections. Okay, and if you see whatever peptides we have designed in computational part from the genome and other databases and tools, we can directly synthesize in the peptide laboratories, and these peptides can be tried with in vivo experiments. That is in preclinical studies. In preclinical studies, we can try with the computationally developed sequences. Okay, so after trying with these peptides, we can also understand the molecular epidemiology. There is some two important terms: epidemic, pandemic. Epidemic is pertaining to a certain region. Myself, I worked on a research and I also received multiple grants. I worked on a type of virus called Nipah virus. Have you heard about this Nipah virus? So the second outbreak happened in 2018 in Kerala. So it claimed around 18 lives. Okay, just 18 lives. But if you see SARS-CoV-2, millions of lives. The reason behind how they curved the kind of fatality, how they made it. So this Nipah virus is actually we have one strain. Unlike hundreds of strains, we have only strain, so we don't have any mutation with the Nipah virus. So whatever the one strain infected from the first patient to the second patient, that was immediately diagnosed. Just because they diagnosed and they found the virus is a Nipah virus. Okay, well, I think after sacrifice of 4 to 5 lives, a doctor called Ashok has come up with. He's actually an US virologist. Fortunately, he came to Kerala for vacation. During that time, when the patients approached, he identified that is something new. It's not like a regular kind of a viral infection. Later, after identifying it, so at least they can able to save this much lives. Okay. So for the Nipah virus, what I worked is I worked on both B cell epitope and T cell epitope, but for the B cell epitope already the papers are published. So I tried to publish the T cell epitope. Okay. So molecular epidemiology now we understand in the Kerala alone we can find this infection in that particular year, and now it was totally abolished. Okay, so this infection we don't find any patients with this infection. So initially, whatever peptides use, okay, we try to initially treat with healthy volunteers. Directly, we won't treat the disease volunteers. Initially, we go with the phase one trials with 100 healthy volunteers. Later, 2000 scale up to 1000 healthy volunteers in phase two trials. Later, we treat with disease patients. The disease patients, whatever we treat, will be 1000 and more than that in phase three. So, and if it comes to the FDA approval, post-marketing surveillance, obviously it has crossed the phase four track. Now, if you come across the reverse vaccinology, the first vaccine discovered from reverse vaccinology is meningococcus B vaccine. This is used to treat bacterial meningitis infection. Bacterial meningitis infection. Okay, so if you see this kind of meningococcus V vaccine, how it is discovered from reverse vaccinology. Initially, from this bacterial meningitis, okay, which is infecting the brain, they have taken and when they sequenced it, they find able to find 600 potential vaccine candidates or identified. Later, whatever that potential vaccine candidates, that is protein targets they identified, they try to uh, use the recombinant technique for scaling up the production. When they tried, only 350 proteins out of 600 expressed in E. coli. And in the next screening, they tried checking with novel surface exposed proteins. That is, the surface proteins present on the cells. They identify only 91 out of 350. And out of that, 28 novel proteins have bactericidal activity. So these 28 novel proteins are taken, and from that, the epitopes are discovered. Whatever epitopes are discovered, and whatever we link a multiple epitopes to form a multitope that is used to treat at present the to treat this meningitis infection. You know, at present for the meningitis, this is the best vaccine. That is multitope based vaccine. Multitope, I mean multiple peptides connected to each other. So this is actually this question I asked in GAD. Okay, what is the first vaccine discovered from reverse vaccine? Now let me take you to the next thing. A publication which happened in 2020. Okay, so I just published this part T cell epitope based vaccine, and this also only two projects selected as best projects in India. So of course this is one of the projects. Okay, and this also we received funding from Tamil Nadu government. TNA-XXX. So now let me explain about the workflow. So we initially started with a type of protein called protein W. We have so many structural and unstructural proteins in Nipah virus. Whatever other structural and other non-structural proteins, all these are already worked and published. So the only protein the, yet to be published is protein W. So only I've chosen this as a tab. So I've taken this pasta format of the sequence. Of course, this is also a virulent in nature. So I've taken this protein W as target, and I started identifying the T cell epitopes. Okay, after identifying the T cell epitopes, then I screened the best epitope out of thousands of epitopes whatever is generated from this protein W. So out of 1,000 epitopes, whatever the best epitope I mentioned, this I finally docked with HLA molecule, MHC molecule. And then we try to, after docking, we go on with the MD simulation to check the stability. If molecular docking is used to check the binding affinity in terms of energy in kilocalorie per mole, in molecular simulation, we check the stability of peptide. This HLA molecule present in humans actually. HLA molecules are usually present in humans, and whatever peptides we have designed, the source is taken from Nipah virus protein W. So this is from a Nipah virus. Okay, the Nipah virus source, we try to talk with human HLA. Then only we can come to know whether this peptide uh, is going to treat. Is going to prevent these viral infections or not. There is much more immunology. If you go to the subsequent slides, you can understand better. Also, one more example I can provide, that is SARS-CoV-2. In SARS-CoV-2, we have totally 16 non-structural proteins. In non-structural proteins, we can also find proteases, polymerases. These are, of course, most important proteins in other viruses. But when compared to the non-structural proteins like proteases and polymerases, the most important target we find in SARS-CoV-2 is nothing but a structural protein called spike glycoprotein, which is present on the surface. Okay, this is the corona-like shape, so we call this as coronaviruses. And if you see this spike is one of the structural proteins. Then what are the other structural proteins? Envelope, membrane, nucleocapsid. So totally 16 non-structural proteins and 4 structural proteins are present in SARS-CoV-2 gene. So like this, I can see in Nipah virus also. Okay, I just mentioned you the structural, non-structural part. Now let me take you with another example, like how to treat this SARS-CoV-2. How to prevent SARS-CoV-2. If you see the vaccines, everyone know about Covishield and Covaxin. Can we discuss more about that? The Covishield, I think many of them have taken Covishield. Next to that, you have taken the Covaxin. So my question here is, what is the efficacy of Covishield and how about the efficacy of Covaxin with the first alpha strain of SARS-CoV? Efficacy, I mean, how many percentage of people get prevented from these viral infections? Do you know that? So you know
Sir,
that? Oh, yeah, yeah. Please, ma'am. Please, please.
sir, they said Covishield is better than Covaxin that time, sir. But exactly that number or percentage, I'm not aware, sir.
Ah, that I can mention, ma'am. Very good. Thank you. Actually, this type of interaction is what I am anticipating from others also. But practically, when we explore to the literatures and other kind of a data, we came to know that in both the vaccines, we use primer dose and secondary boosters, isn't it? Primary and secondary. So two doses are taken. So the two doses are taken just to increase the bioavailability and even the efficacy of whatever vaccines we have taken. So if you see the Covishield, the efficacy is coming around 83 percentage. And if you see, this is after the second booster. But the first dose, whatever we take, okay, it's not crossing more than 45 percentage. But whatever 45 percentage efficacy, if you want to improve further, okay, so if you take the second booster, it comes around 83 percentage. That is 17 percentage of population can't still able to prevent from that SARS-CoV-2. That's what it infers. Covaxin, it come around, the efficacy came around 92%. It depends upon individual to individual. For India, it might be better. But if you go to the US or other kind of countries, the efficacy can still decreases because the vaccines are designed to treat specifically Indian population because these are manufactured in India. Similarly, if you take this kind of a vaccine called COVAX, this is a multitope based vaccine. I mean, we connect multiple peptides in this vaccine. This is generally, it is a product of US. So, the efficacy? The efficacy is more than 99.9 percentage. Means that most of the people get prevented from this SARS-CoV-2 infection. So how is this possible and how is this constructed? Initially, they have taken five specific cytotoxic lymphocytes, okay, and or including the T helper. So in T cell epitopes we have types of epitopes. One is CTL, the other is TH. CTL stands for cytotoxic T lymphocytes. The whatever TH represents helper T cells. Okay, so from these two subtypes of T cells, five specific epitopes are taken, linked with the TH epitope, that is helper T cell epitope, and this also linked with the CpG1 nucleotide. And further, here in the right side, you can see the adjuvant is linked. The adjuvant they have used is the aluminum phosphate, okay, which is inorganic in nature. And together, these six peptides, along with the CpG and adjuvant, is attached with most important fragment, that is receptor binding domain of spike glycoprotein. Receptor binding domain, in SARS-CoV-2 spike glycoprotein, we have around 1,200 amino acids. Okay, and out of this, the approximately 1,200 amino acids, okay, only 13 amino acids are the binding sites. And whatever the binding sites spanning this 13 is what we call it as receptor binding domain. So this receptor binding domain of a fragment of spike glycoprotein is linked with these peptides, and these can able to yield around more than 99.9% efficacy. I think many of them didn't hear about this because we don't have this vaccine in India. Now, if you take this comparison on treatment of this SARS-CoV-2, okay, you know that monoclonal antibodies are very highly specific. They are target specific, especially for treatment of cancer. The best treatment to prolong the lifespan is monoclonal antibodies. For treatment of cancer, we generally like if they are advanced stages, in chronic stages. In chronic stages, this is the only treatment, monoclonal antibodies. But if they are in initial stages, okay, we can also try with the small molecules for treatment. And if you see this kind of a comparison for SARS-CoV-2, the safety aspect we can find with COVAX is showing the excellent profile. And if you see, it is more stable, it is simple, scalable, cost effective, and strongly existing, and it has having high potential. When compared to monoclonal antibodies and small molecules, these peptide-based vaccines hold the best for prevention of these viral infections. So only we are discussing on specifically this reverse vaccinology, which is majorly can find the product is nothing but peptides, okay, from the viral sources or bacterial sources, whatever we want to develop, okay, for which we want to develop. Now, let me take you to the T cell epitope. So in T cell epitope, I told you there are two types of T cells, cytotoxic and helper T cells. Generally, if you take this kind of a T cell, which is having a CD4 receptor on the surface, so whatever the orange color you can see, this is the CD4. This is a receptor present in the CD4 T cell. So this along with the T cell receptor, which is shown in green color, if it interacts with the MHC molecule of APC. MHC molecule is generally present in APC, where you can find this region is nothing but a peptide. If a peptide is generally presented on the MHC of APC. I also told you the example of APC. The example of APC is B cell, dendritic cell, macrophage, natural killer cell. All these things comes under antigen presenting cell. Okay, all these things comes under antigen presenting cell. So this if it interacts with the T cell through CD4 and T cell receptor, the T cell gets activated. If one T cell gets activated, two T cells are formed, as I mentioned earlier. Even the proteins will be released. Similarly, there is another category of T cell where it has only CD8 on the surface. Instead of CD4, we can have CD8. But the same process, CD8 with the T cell receptor interacting with the antigen of MHC molecule. But here we have MHC2 molecule. Okay, CD4 is generally interacting with the MHC2 molecule of APC. But here in CD8, it is going to interact with the MHC1 molecule of APC. That is the only difference. The one difference is if it is a CD8, it is going to interact with MHC1. If it is CD4, it interacts with the MHC2 of APC. Now, let me take you further with another cell epitope, that is B cell epitope. Even in B cell epitope, we have two subcategories of epitopes. The first one is the linear or continuous residues epitope. Here, if you see this epitope, which is from the antigen part which is binding with the antibody is what we call it as epitope. If this is epitope, whatever the antibody region you can find, okay, which is binding with the epitope is what we call it as paratope. Okay, the paratopic region is present in antibody, generally interacts with the epitopic region of antigen. So here in this linear or continuous residues where I can say all the amino acid residue 19 to 23 in continuous, we need just the sequence of epitope and the structure of protein to bind. But whereas here in this epitope, We need the structure instead of sequence. If you have a structure, see, a part of an amino acid of a peptide is going inside, and so it's going to have a high affinity. Whatever the epitopic of antigen is going to bind with the paratopic region of antibody, just because it's going inside and outside, you can see which is having a good affinity when compared to this continuous residus linear epitope. Just because we have having one amino acid is going inside, okay, we can find it is a discontinuous residues in nature. You don't have a same length, okay, connecting one amino acid with another amino acid. So this we call it as conformational epitope because this is structure based prediction. Now let me take you to the next B cell receptor, T cell receptor. B cell receptor present in B cells, T cell receptor present in T cells. Okay, so this T cell receptor along with the CD4 reacts with the interact with the MHC2 to make a T cell activated. You remember? Similarly, we also have B cell receptor. This is useful when we go with the B cell immunity. Now, something about the types of cells. Earlier, I didn't show you what type of T cell is CD4 molecule. So that is nothing but helper T cell, which is represented as TH. The helper T cell has only CD4 on the surface of the T cell. And similarly, cytotoxic T cell is represented by TC. Further, we have subtypes also. Even in T helper cells, we have TH1, TH2. We have subtypes also in helper T cell. Similarly, in cytotoxic T cell, we can able to see CD8 molecule is present on the surface. Okay, so let me take you to the next slide. My PhD research. In my PhD research, I majorly worked on dendritic cells. In dendritic cells, I've taken around 162 target proteins, surface proteins of dendritic cell, and then I uh, have worked on dendritic cell stages of development. Dendritic cell initially will be in immature dendritic cell generated from monocytes. This is an immature dendritic cell. Generally, you don't have sharp arms like this. This is actually a mature dendritic cell. It looks like a starfish, isn't it? So initially, the immature dendritic cells are generated from monocytes, which is round in shape. From monocytes, the immature dendritic cells are developed, and from immature dendritic cells, the mature dendritic cells are developed through maturation process. If you see this dendritic cells, okay, we have molecules like MHC1 as well as MHC2. If it is, we have a MHC1 molecule on dendritic cell, it interacts with the CD8 molecule of T cell. If it is having MHC2, then it interacts with the CD4. Similarly, what are the other interactions important for T cell activation and dendritic cell activation? So, if you see the next important molecule is the co-immunostimulatory molecules like CD80, CD86. Okay, so these are co-stimulatory molecules present on dendritic cells. If it interacts with CD28, what is going to happen? T cell gets activated. Apart from interacting CD4 or CD8 with MSC2 or MSC1. But instead of CD28, we also have a receptor called CTLA4, cytotoxic T lymphocyte antigen 4. If CTLA4 instead of CD28, if CTLA4 is interacting with the CD80 or CD86 molecule, then instead of T cell gets activated, the T cell is going to get suppressed. Okay, it's going to suppress. Like generally, if you want to combat cancer, we need T cell to be get activated because one T cell going to proliferate into two T cells. If you have more T cells and dendritic cells, then we can combat cancer easily. But this inhibition or suppression is going to happen when another molecule surface receptor CTLA4 interacts with CD8. So then, where can exactly we use these kind of immunological things? This we can able to use for treatment of autoimmune disease. For treatment of asthma, psoriasis, arthritis, we can use immunosuppression, whereas immunoactivation in immunomodulation can be used to treat cancer. Got it? For this kind of invention, okay, the Nobel laureates have received uh, the Nobel Prize, I think, on 2019. And the father of dendritic cells, Ralph Steinman, received Nobel Prize on 2014. So, plenty of Nobel Prizes are there in these kind of immunological interactions. Also, third, fourth, we can also link somehow with these kind of cellular immunity. Okay, because cellular immunity is also very important for prevention of viral infections, prevention of cancer and other diseases. Now, I'm going to explain you the mechanism of how the antigen is processed and presented on the T cells. I told you, I mean, on the APC, sorry, the antigen is generally present on APC. APC is nothing but, I told you examples like dendritic cells, B cells. In this case, the cell is looking round, so it is a B cell. If any of the virus is going to attack the B cell, okay, the B cell, I mean, that virus is going to degrade it through proteasome. So proteolytic degradation is going to happen with that viruses or bacteria which is going to inside, and so the proteins, whatever getting inside the proteasome, is going to degrade into small peptides, and these peptides are transported through transported associated protein present in the endoplasmic reticulum organelle, and these peptides will be transported further through the MHC and presented on the surface. Finally, it will be transported through Golgi complex and then further transferred through secretory vesicle, and finally the MHC molecule is going to present on the surface of APC. If T cell interacts with the antigen of APC, then the T cell gets activated. So this is the natural process going to happen. So this is actually MHC1 antigen processing and presentation pathway. So why I'm telling you, actually tomorrow I'll be taking you more than 50% hands-on. Okay, so but if I take your hands-on, just putting a sequence, getting an output, but you don't know what is exactly present in output. To know more about understand the immunology, one should understand all this. Okay, these are the basics I am covering. The results you find all this, but you should know what is the basic. So I'm teaching now. Now let me take you further on the structure of MHC molecules. In MHC molecules, we have class 1 and class 2, which are the major molecules. We also have MHC class 3, but generally that is of minor importance because that is used for complement molecule activation, which is minorly considered with respect to immunity. Now, if you take this MHC class 1 and MHC class 2, so you can see the shape differences that is on the surface of antigen where it's going to bind. The MHC class 1, you can see alpha 1, alpha 2 membrane distal domains are present on the surface. That means we have a same subunit, alpha and alpha 2, or comes under same subunit. So, whatever antigen I'm going to bind, okay, is of lesser length. The usual length we use for predicting is 9 amino acid for MHC class 1. But whereas if you see class 2, we have different domains on the surface. Alpha 1 unit is different, beta 1 unit is different. So only you can find the gap here. So we have to increase the length of the peptide from top to bottom, like how you paste the sellotape. If you have a two different thing, okay, increase the size of the serotonin, isn't it? Similarly, here you increase the size of the peptide. So additionally, you have two amino acids, and here additionally you have two amino acids. So 9 plus 2 plus 2. So obviously we choose 13 amino acid to 15 amino acid. So 13 amino acid to 15 amino acid for MSC class 2. The peptides, whatever we predict, like epitopic peptides, whatever we predict, we have to fix a length. This is 13, 9 amino acid, and this is 13 to 15 amino acid. So instead of 2, if you have 3, then obviously we have 15 amino acid because here also we have 3. Now let me take you further on MHC genes and function. If you see the MHC genes, okay, we have majorly a class 1 MHC genes and class 2 MHC genes. What is MHC? I told you it is major histocompatibility compatibility complex. This is what we call it as MHC. Why we are specifically bothering on this human MHC molecules? You know, the MHC is generally we call it as a protein name, and this has a type of genes called HLA genes. HLA stands for human leukocyte antigen genes. Each and every human distinguish not only with a fingerprint, but they also can distinguish with HLA gene signatures. In HLA gene prototyping, you can see even the twins have different HLA genes. If twins itself, the born twins itself have different HLA genes, obviously each and every person distinguishes with this. But if you take other proteins in humans, for example, the hemoglobin gene of yours is mine. I mean, yours and mine is same. And even if you compare with others, it will be same. So most of the proteins in humans share the common genes, except these kind of MSC or HLA genes. So the treatment of or a prevention kind of a thing, that is a vaccine design for a human, varies with person to person. So not only the drug we have to tailor according to the specific patient gene, but also the vaccine, whatever you use for earlier prevention, is also has to be designed and developed for each and specific to each and every person. Got it? Now let me take you something about class 1 MNC genes. These are generally nucleated cells, and major function is to present the peptide antigens. And generally, MHC genes, okay, is present peptide antigens to only cytotoxic T lymphocytes. Cytotoxic T lymphocytes, what do you mean by that? It is going to have CD4 or 8. It is CD8. Cytotoxic T lymphocytes have CD8 on the surface. If it is class 2 MHC genes, okay, then it present peptides on T helper cells, which have CD4. You know, the HIV patients during the dying stage, okay, can see the CD4 population the count is zero. The HIV patients have CD4 count zero when they are about to die. Slowly the CD4 cells will be dying, okay, if they are infected with this virus. Class 3 MSC, this is actually respond to the complement system function. Now, if you see the difference between MSCs, there are plenty of differences, and if you want to know more about this, you can study the textbooks like Immunology by QB, or Immunology by Freud. These are the preferred textbook to study more information about the MNC molecules and its differences. Now let me take you, because whatever I am showing this is important for practicing. For hands-on, we have to know all this. Whatever I am skipping, that is just up to your knowledge point of view. Now let me take you to the next thing. HLA MHC complex. The HLA genes, I told you it varies with person to person, including born twins. So under that case, these HLA genes are generally present in chromosome 6. We have 22 autosomes, and X and Y are 6 chromosomes, totally 24. The total we count is 24, which is going to comprise a genome. Okay, but only one chromosome, which has all the genes, is chromosome 6. It has both the MHC1 as well as MHC2. The major MHC1 genes are HLA-A, HLA-B, HLA-C. Similarly, the major MHC2 genes are HLA-TP, HLA-TQ, HLA-TR. Remember this TP, TQ, TR, ABC. And the other thing, whatever mentioned are the minor chains. Rarely we find this chain, so can ignore. You can also see the locus regions of these genes, HLA-ABC and HLA-TP-TQ-TR. Because I'll be showing you on tomorrow about all this directly to the in the website. Now, let me explain you something about the database what I'm going to provide demo on tomorrow. If you want to practice with the T cell epitope prediction and even B cell epitope prediction, we can try with IEDB website. IEDB stands for Immune Epitope Database. Okay, so we can also see the historic development of vaccine. So in my lab, in my research lab in SVC, we are working on the latest adjuvants like AH04, AH03, IL4. In my PhD research, I worked in all these adjuvants, whatever I mentioned. That is the interleukins, granulocyte macrophage, colony stimulating factor, and saffron and other plant-based adjuvants we have tried in our lab, including we can also use the dendritic cell. I told you dendritic cell is an professional antigen presenting cell. This also acts like an adjuvant. Not only the protein molecules acts like an adjuvant, even the cell itself can act like an adjuvant. Okay, so in this era, the researches happened in that time, and even myself I worked in that. The most popularly, I mean, for more than 100 years, if you see the most popularly used vaccine adjuvant is alum. Okay, we have different aluminum salts like aluminum sulfate and aluminum phosphate for which can be used as a vaccine adjuvant for different vaccines. Now related to the database what we have developed in SVC in 2017 and 2018, two different PG students have developed this kind of a tool. So that we named as a immunoanalytical database. You know what is the specialty in this database? In this database, if you want to uh, find which is the best plant compound for the treatment of any autoimmune disease or any cancers, we can able to find. Okay, for this, we have talked more than 1,000 interactions, 1,000 molecules, plant-based molecules with the dendritic cell receptors, and we have posted it. Whatever we talked, we for talking, we used a commercial tool, Discovery Studio. So Discovery Studio, the CanFit we use for talking, and these results we have incorporated through SQL, through PHP. So this we made it in C# platform. So for coming up with a kind of a best plant-based vaccine adjuvant for a specific disease, we have developed this. And this is something about my PhD research. In my PhD research, I worked on plant compounds like vitamin B and vitamin A. So vitamin B is derived from a, uh, a plant called Verivia diffusa. In Tamil, we call it as Mookkaratai Kirai and Withania somnifera ashwagandha. Okay, from that we have taken vitamin A. So these vaccine antigens, BCG, tuberculin, tetanus, toxoid, we have procured without antigens and we have tried with these adjuvants and we try to check whether the plant-based compounds can differentiate, the initially, if you want to develop the immature dendritic cell, I told you we have to develop from monocyte. This is what monocyte. So from monocyte, immature dendritic cells are developed. If this is a kind of a differentiation, this process of development we call it as a differentiation of monocytes to immature dendritic cells. The next process to come is after the immature if you want to bring it in the form of a mature dendritic cell, we have to undergo a process. I mean, the cells have to undergo a process called maturation. So we have checked whether these plant compounds can involve in differentiation and maturation. Fortunately, we find these plant compounds also play an important role in both the things. So whatever vaccines we develop is more efficacious on treatment. Okay. So this is more of immunology is there. Like using these plant compounds, we can produce more antibodies. We can also produce more T cells. Okay, cellular immunity are also increased. Okay, I've completed my session today. So full of theoretical session, isn't it? So tomorrow I'll be taking much of practical session. Okay, so try to join on tomorrow before 7 p.m. So after that, Sunday is a break. Okay, and we have, I believe we have 2 or 3 more sessions left out to complete this course. And for the MCQ alone, instead of completing on the same day, I can give you some 4 or 5 days. I gave some 4 days time. Okay, from the day of completion, I can give additional 4 days time to complete the MCQ so that if you have missed watching any videos, you can watch the videos and you can present it. Okay, still whatever you are going to complete the MCQ is merely for self-assessment. The marks are not going to reflect. Okay, so till now, if you see whatever courses I've conducted, the average score is more than 85 to 90 percentage in each and every course. It means that everyone are trying best on acquiring the concepts. I believe even you can maintain the same. Okay, so we have at least one Sunday gap. So utilize this time and complete even the MCQ and overall feedback, which is important to secure the certificate to your email ID. Okay, if you complete overall feedback, which I'll be separating on the last day, and the MCQ, I'll be providing you the certificate. Okay, I've completed today's session, a theoretical session on what is immunoinformatics and what is reverse vaccinology and the immunology part. Any doubt you can ask me at present in this. If you don't have any doubts in the theoretical part related to the practical part, we see on tomorrow. Good night to everyone.
Thank you, sir.
Thank you so
Thank you.